Welcome to the 54th InterScience Conference on Antimicrobial Agents and Chemotherapy, or ICAAC, and this is ASM Live. I'm Michael Schmidt from This Week in Microbiology, and together with the help of the program committee, we've selected some of the science that's being presented here this week and asked the authors to come and discuss with us their exciting results. So welcome this morning with me. I gotta find the names. Laurent from the University of Fribourg in Switzerland, and Karim Morey from the Oregon State Public Health Laboratories. Today we're going to learn about tests for the rapid detection of antibiotic-resistant bacteria. If you're like me, every morning when you open the newspaper, even in a city as small as Charleston, we learn about the scourge that antibiotic-resistant is wreaking havoc in all of our hospitals. Today we're going to learn about the rapid detection of antibiotic resistance and how it is vital in assessing the appropriate antibiotic therapy for infections. And the participants, Dr. Poirel and Dr. Morey, are going to share with us the data of and underscore this word, two new inexpensive tests to detect antibiotic-resistant markers within hours or even minutes. And if you tuned in yesterday and heard our discussion on antibiotic stewardship, you will immediately appreciate how important today's discussion is in dovetailing with the discussion on stewardship. So welcome, and thank you for agreeing to do this. So we're going to start with ladies first. So why don't you tell us a little bit about your exciting results?
Sure. I work for public health in Oregon, and we started to have an initiative to prevent the arrival of carbapenem-resistant Enterobacteriaceae, usually known as CRE. And as a result, we started with a modified Hodge test that I believe everybody is familiar with.
What is a modified Hodge test, that just so the folks at home can follow along? You know, the acronym soup to the members of the audience is old hat, but to those listening out in the Twitterverse, it's something to be of concern. So just help them to figure out what's going on.
The modified Hodge test, it's a phenotypic test. It's just characteristics to make sure that is it there or not.
So it's a yes or no answer.
And it was sensitive, but it was not specific enough. It was very subjective to interpretate, and we would have an answer at the lab and another answer at a different lab. So we were looking for a better test that was faster, that was more sensitive, more specific. So we came across the Carba NP, which Dr. Norman, Dr. Poirel, and Dr. Dortet came up with in 2012. So it looked promising, it looked highly specific, highly sensitive, and a quicker turnaround time.
One of the things that my dear medical students always call me to task for is what's the difference between sensitivity and specificity? And my shorthand answer is sensitivity are true positives, one that has the antibiotic resistant trait the test is asking about.
Correct.
And then specificity is true negatives, where they don't have the ability to become, they don't have the antibiotic resistance marker presently. So when I tell them true positives and true negatives, because those two words begin with S and they confuse them. So again, I always bring that up just so that we're all playing from the same field, so to speak.
So after evaluating the test, it worked really well, just as it was described. We evaluated the method that the Mayo Clinic was currently using, and it was also slightly modified from the original protocol by Dr. Norman and Laurent and Dortet. And it worked, but our sensitivity was 92%, whereas the original protocol showed 100%. Our specificity was very similar, 100%, and It's fast, like I mentioned before. It's yielding the results that we were expecting, and as being fast, it's also allowing the physicians to intervene right away instead of waiting another day for results.
So that means the physicians, once you're able, in your case, the public health lab, you're able to issue a finding that will help them change the care of the patient.
Correct.
So they're just not giving antibiotics that do no good.
Yeah, and not only it's helpful for physicians, but it also for epidemiology reasons. So once they know, our epidemiology section is notified, and they start all the coordination to isolate this patient. And if there's been any contacts with other patients, then we will perform a screen for those, so it doesn't continue spreading to other populations.
Dr. Poirel, share with us your work. So you actually have 2 posters here, so we're going to get double duty out of you here this morning.
Indeed, yeah. We have 2 posters on this so-called ESBL test, and we had some other 2 last year on the Carba NP she was referring to.
So ESBL is extended spectrum beta-lactamase, and Carba is carbapenemase, the markers that you're effectively both looking at.
Indeed, because nowadays there are 2 problems. Of course, we know that there is a kind of endemicity nowadays of ESBL producing enterobacteriaceae. So those isolates, they do produce these enzymes, these extended spectrum beta-lactamases, and as a matter of fact, all those isolates are resistant to broad-spectrum cephalosporins once they acquire that kind of enzyme. And it's a critical issue everywhere in the world, and we have to identify as early as possible all patients who are carriers of those bugs in order to prevent further dissemination in hospital settings and so on, and also to adapt the treatment. And then there is another step, which is the production of carbapenemases. And those carbapenemases, they do also hydrolyze all cephalosporins that you usually have as a treatment, a first-line treatment. But also they include in their hydrolytic spectrum the carbapenems that usually are the gold antibiotics that you can use at the last resort once you know that the patient is infected with an ESBL producer. You cannot use cephalosporins anymore. You know that you can switch to carbapenem, you can save life that way. The problem is, again, that now we see and we are facing out the emergence of these carbapenemases all over the world. And that's what we call the nightmare. And
it is the nightmare scenario.
This is a nightmare, yes, because you end up with patients you cannot treat anymore. You do not have any drug you can use safely for treating these patients. So then you start combining different antibiotics. So
you mix and match.
That's the trick. So our idea, Professor Nolman and myself, was to develop a test that could be affordable for everyone.
And that's the key from your selfish perspective in public health.
Correct.
Is you really want to be able to do the epidemiology, and the only way you're going to be able to afford it is if people like Dr. are able to reduce the cost.
Yeah.
Correct.
So to reduce the cost, because if you want people to use the technique, of course, it has to be costless.
Cheap.
Cheap.
Cheap's the ugly word. But
okay. First thing. Second thing, to be rapid. Why should it be rapid? Because you are dealing with infected patients, so you have to adapt the treatment as soon as possible. And second, it has to be quick also in terms of infection control. You have to recognize who are the carriers in your hospital. So if your infection control team wants to act as early as possible, and you have to, as a clinical microbiologist, you have to give them the answer very, very early.
And we recognized in the United States that we had an outbreak of these evil ESBLs, and a hospital actually closed and demolished the entire ward where the ESB was, and then repainted everything brand new, and boom, within a few weeks it was back.
Yeah.
So again, your test could add utility.
So this test, we thought about first starting to develop this test when you pick up the colonies. They grow after 24 hours, so you have the clinical specimen, then it grows, and those colonies growing on the plates, you want to know whether they could be resistant or not. And one quick way to know whether they could be resistant or not is to identify whether they do produce those kind of enzymes. If they do produce enzymes, very likely that's going to be resistant to these antibiotics. And the way around, of course, if there is no production of that kind of enzyme, you can probably extrapolate that those isolates are going to be susceptible to antibiotics. So we decided to develop this test from colonies, and it worked very well. So on one side, we have the detection of ESBL production. So in that case, we use a substrate which is cefotaxime. Cefotaxime is a third-generation cephalosporin, and the idea is to put together the antibiotic on one side and an extract from the bacterium on the other side. So you pick up the colony, then you just perform lysis, and then from this lysis you have the enzymes. So if you have the enzyme and you put together in a cube with the antibiotic, you're going to have hydrolysis. And the idea was to have something that could trace this hydrolysis. So to have red phenol, this is what we use as a colorimetric reagent, was the solution we found out that was the best. Because once you have hydrolysis of the antibiotic, you have decrease of the pH. And
this is old-time microbiology.
Exactly. So this is a very simple, basic microbiology. Yeah. And so if you have hydrolysis, you have acidification. Acidification means reduction of the pH. And so this red phenol is going to turn from red to yellow when there is hydrolysis. So we found that that technique could be very useful. So adding cefotaxime as substrate to detect extended spectrum beta-lactamases. And on the other side, we thought about also doing the same for carbapenemases. And in that case, the substrate is the carbapenem. And in that case, it was imipenem. So we had these two techniques, and we published that last year, and many groups now all over the world are using this test technique, which is named Carba NP. The NP are the initials of my boss and myself.
Yes.
And because we found it funny to advertise people about that. And we said, okay, let's go like this. We can pick up the colony, and in few minutes you can know whether this colony is going to produce an ESBL or a carbapenemase. And the second step was then to think about the clinical specimens and to have the possibility not only to wait these 24 hours to pick up the colony on the plate, but why not trying to figure out, at the beginning from the sample, whether you could have that kind of bug inside and if those bugs are producing these kinds of enzyme or not. And so this is what we worked on the last year, and that's why we are presenting this data now at the ICAAC, to have the possibility to test from blood cultures on one side and from urines on the other side.
Urine. So if I was a patient, which would I rather give to the healthcare provider? Someone who's going to stick my arm or fill this jar?
Yeah, that's not exactly the way it works, because you have suspicion of urinary tract infection. In that case, you're gonna give urine, and if there is a suspicion of septicemia, of course, this will be the blood cultures.
But in the same line, not to trivialize it, urinary tract infections are, of course, a substantial hospital-associated infection, and it's a significant problem all over the world.
Yeah.
And CAUTIs or catheter-associated UTIs, if you can get at that issue with your test, that would be phenomenal.
So it was very easy to adapt this test for urine or for blood cultures. As soon as the urine is considered as positive in a clinical lab with 10 to the 4 leukocytes per milliliter, and we see with gram staining that there are some gram-negative rods. In that case, there is a high suspicion of infection. So we just pull down the cells, and then we use the same lysis buffer, and we perform this test. And in something like 20 minutes, we can have the possibility to answer to the question, is there here an ESBL producer or not?
So from a public health laboratory's perspective, is this a good thing?
Well, it is, because everything is focused on detecting this type of infections or bacteria as soon as possible, and reducing the turnaround time, like I mentioned before, and a rapid intervention for physicians and infection control.
Now, given the utility of these two tests, have you begun to encourage the laboratories that feed data to the public health system to adopt these tests at their benchtops rather than you having to do it?
Correct. We started by the Carba NP test, and we are the reference laboratories for the clinical.
For the state of Oregon?
Yes, for the state of Oregon. And basically, they have all their automated ID and susceptibility systems, and when the results for susceptibility meets our criteria to consider a potential CRE, that's when they submit the isolate. So they have thought about implementing the Carba NP test, but I know there's some financial limitations or staffing limitations, so that's why as a public health, we had the responsibility to continue monitoring for those. And they are currently submitting those isolates for us to confirm. And after Carba NP, we implemented the PCR test for final confirmation. So we want to do that full testing to have a complete result.
So in Switzerland, what are they doing with your tech?
Yeah. So they use this technique routinely nowadays, and we are used to report to clinicians because we know that it is reliable. So that's also, we were talking about sensitivity and specificity, and when you achieve very high rates for both of them, you can say that the test is reliable, and it is. And we also, Professor Norman and myself, we are still also working in a hospital in Paris, in France, and there it is now commonly used routinely that this test, they rely on this. So as soon as you get the answer, there is a production of an ESBL, the clinician, they switch to carbapenems because they know that this strain is going to be resistant to cephalosporins. So they implement the adequate treatment very, very early. And that's the best way to treat patients, of course. And on the other side, the infection control is so happy with that. You cannot imagine every time they call and they say, okay, you can tell me in a couple of minutes whether this patient was colonized with a carbapenemase producer or not. So that means they know whether they have to isolate the patient or not.
So when you take, for example, the urine that you have the suspicion, you lyse the cells, you add the substrate, which is the antibiotic, and you then ask the question, does the pH change? How fast does that occur? Minutes?
So the color change, usually it's something like 5 minutes, 10 minutes for most of the enzymes. There are some enzymes with a bit weaker hydrolysis capacity. In that case, it can take something like 1 hour, but never more than that, never more than that. So that means very short delay between you get the sample and then you give the answer.
So are there any other confounding enzymes that could similarly reduce the phenol red from its neutral pH to its acid pH?
So actually there is a control every time in our test to see whether there is no color change without any beta-lactamase activity because we need that kind of control, of course. Actually, in my hands, it never happened that we had some bugs with that kind of false positive and same.
Well, hence this true positives, true negatives.
Exactly. So that's again why we can consider this test very reliable.
You're watching ASM Live, and I forgot to encourage those of you out watching on the internet that if you have a question, feel free to post it either via Twitter or our open chat line. Similarly, the audience, we're open for questions, so if you have one, please attend the microphone and ask your questions. Again, we're visiting with Dr. Laurent Poirel and Dr. Karim Morey from Oregon State's Public Health Lab. We have a question from the audience.
Ed Sussman with MedPage Today. Are these tests now approved? Are they available commercially in the United States? Has the FDA given their okay on them?
So this test, we got a patent on this, and that the license has been purchased by a company. It was just something like 1 year ago. And the product should be available on the market starting from November the 1st in Europe, next November in Europe. And according to what I heard, that should be 1 year more for the US.
Okay, and have your studies actually been done on patients that come to the hospital now, or are you still just using laboratory trials?
So we do both, actually. We do both. We decided, in our hospital in Paris, we decided to adopt this technique routinely. And so we do that.
Okay, and what's being done in Oregon now? Are these tests being used
diagnostic
for
purposes.
diagnosis
Only-
purposes in patients?
Only on clinical isolates from patients.
So you're not really using them as you would expect it to be used once these tests are approved?
We haven't made the leap to begin to use them into the standard of care, where you would change the drugs.
Right, it's still non-FDA approved. So whenever we result that, it has that message that it should not conduct any final decision for treatment. It's just a reference.
But I just want to know if you're testing actual patients, or you're just still testing isolates?
Yes. Yes.
So are
Which
you
one?
testing patients or not?
Yes.
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 Depends what you call it.
Yeah.
What you define
Well,
as patient.
they isolate.
My real question is, have somebody come in with an infection, you've given them this test, and you've determined whether they have a resistant bug or not?
Yeah.
In the United States?
Well, that in Oregon, it starts by the reference lab. And then once these other labs determine if it meets the criteria or not, then they submit it to our lab. And then we screen it to see if it's Carba NP positive or negative.
So my real question here is it being used in patients today? If a patient comes into one of your hospitals in Oregon today with a suspicious infection, do they get this test? Or is this still an experiment?
No, they do get the test. Yes. They are required to submit any isolate that meets the criteria.
And are they treated on the basis of this test, or do you have to go through all this other system first before you get a decision on how to treat?
That depends on every institution, whether they want to implement the same line of treatment or a different one. Our job is to produce results and then report it to the institution that submitted that test.
And how long does that take?
The test takes only 2 hours.
But how long does it take to go to you, to go back to them, to treat the
In
patient?
the same day. We test them as soon as they come. We subculture for a fresh culture, and then it's performed the same day.
Thank you.
That's the main difference with the other test, that you need to
Needed
have the
another
colony after
24 hours,
24
yeah.
hours, and then you're waiting for the antibiotic, an additional 24 hours.
Yes.
Hi.
Yes, sir.
Dr. Silo from microbiology in Cardiff, UK. It is clear that you have tested direct urine samples, and has it been used on any other samples? Any?
Besides blood and urine, have you investigated other body fluids?
No, because they are the two main ones that you have to deal with.
All right. Okay, that's all. Thanks.
If your question is related, for instance, to stool samples, because for infection control, you could have the idea to check for in stools. That's something which does not work because it's too much acidic, and so we know already that we cannot use this test for testing stools.
That's why I went back to the false positives associated with the phenol red.
Yeah.
Is you have to understand your specimen in order to have a valid test. To know whether or not your specimen alone will be able to convert the phenol.
Yeah, that's what we do. Yeah,
Yeah.
this is our control. Yeah.
So going forward, if you were the funding agencies, what sorts of studies would you like to see done with tests like this? Do you think they'll have an impact on standard of care? As the gentleman earlier was asking from MedPage, will this test have utility in the standard of care? And what sorts of studies or evidence as clinicians would you want to see in order for you to make a clinical decision to change an antibiotic for a patient?
Absolutely. It will definitely have an impact. And our study just focused on Enterobacteriaceae, but I know it can be used for non-fermenters such as Pseudomonas and Acinetobacter. Once we get there, it's when it's going to have a major impact for them in order to make these decisions. We'll get there, definitely.
So, Laurent, do you think that this test is amenable to automation?
I'm not sure this is the ultimate objective to have that kind of automatization. That this test, the form it will be marketed soon, That would be something like a API gallery. So just pick up colonies.
Microtiter plate, laying it
something
out.
like that. Because you're not going to test it with a large numbers of isolates every day. You do not deal with so many suspicions, fortunately. So the idea is to have this kind of small test, and which will be very cheap. According to what I heard, it should be evaluated between $5 and $10 per test, no more than that. And there is also the big added value compared to other tests, like such as molecular biology and so on. And another advantage I wanted to highlight is that when you use molecular biology testing, you just seek for something you know already. You're searching for genes that have been already identified.
You don't get the unknowns.
The unknowns. And this is always the problem. And there is also a presentation we have here, that's going to be tomorrow, I guess, that we have identified a new Carba penemase. No one knows about that so far except ours. And we know that there are, every year, emergencies of new Carba penemases or new ESBLs. And the big advantage of our test is that since it just detects the destruction of the antibiotic, you don't know exactly what kind of enzyme you have, but this is not what the clinicians want to know, and this is not what the infection
They want to know if it's going to help their patient.
They don't care whether it's KPC or NDM or whatever. They just want to know whether they can rely on the treatment or not, and the infection control team, they want to know whether they have to isolate the patient, to have contact precaution and so on, or that heavy work they have to do or not. So with that kind of test, you do not really consider the nature of the enzyme, but the resulting phenotype.
And the elegance of these tests are, you put in substrate, you ask the question, is the enzyme present, and you get a yes or no answer.
Correct.
Yeah. And then if you really want to identify what kind of enzyme, of course, you use molecular biology, but as a second step.
Yes. We have another question from the audience, please.
Hi, Nicole Chineko from the Public Health Agency of Canada. My question is in relation to the molecular, as well as how you can see your test being used for surveillance. So for AMR or antibiotic-resistant surveillance programs in various countries, whether in Europe or Canada or US. So would you recommend maybe using Carba NP for testing for surveillance programs as well?
Actually, I don't know. It depends if you're rich or not. If you're rich, maybe you can go ahead with the molecular biology and you know that you're going to screen much more than you should have done. But that's not your point if you are rich. If you're not, and this is the case for many countries nowadays, unfortunately, and I know as many of my colleagues performing these epidemiological surveys in Europe, now they like the test because they can screen first and to put out those ones which are clearly not Carba penemase producer or not ESBL producer. So then they go to molecular biology, but only on those ones that will be positive. So it's a good way to save money. Yeah.
Thank you.
Thank you. Looks like we have a question from the chat room.
Yes, we actually have a question from Twitter. Kathleen asks, how does your current test differ from using chromagar?
Good question. Chromagar. Chromagar is, again, a colorimetric assay in which you've added magic chemicals to agar. And chromagar is an interesting approach, but I think at the end of the day, and this is just my bias from being a laboratorian, is chromagar is typically developed for very specific specimens under very tight circumstances, and it's a greater challenge. But this is just me saying this, is the advantage I see in your test is it's very plastic. You can change the substrate, so you can pick your antibiotic that you wish to use and then just simply ask the question, is the microbe have an enzyme that can cleave the substrate? Chromagar, you again load the antibiotic into the chromagar and it will require much more advanced development and much more, a greater degree of validation, I think, than the simple solution-based testing. So chromagar is probably the ultimate. Would you two care to speculate, since this is what you really do for a living?
Sure. Well, I like chromagar for certain groups of testing. And I think
Like
chromagar would be useful for screening. And it would be a large validation, but it reduces the steps. for isolation if there's a carrier patient. Whereas with the Carba NP, we just have a direct testing. We know that it's a potential one, and it goes in 2 hours and it's ready. But
I think it's the speed. Wouldn't you agree?
And not only, when you use chromagar, it's very, very interesting to use that kind of medium because you can pick up those isolates with reduced susceptibility or resistant to some given antibiotics such as carbapenems. But you never know whether this reduced susceptibility is related to the production of carbapenemases or not. And this is something you need to know. In all guidelines, it is recommended to isolate patients colonized with carbapenemase producers, but not with carbapenem non-susceptible isolates. These are the guidelines. So if you pick up something on a chromagar or any other selected medium, you never know. You know that you have suspicion of something which reduces susceptibility, but then you need to proceed further.
There's more testing included.
It's the screening. It's the screening argument.
Yeah.
Another question from the audience, please.
Hi, Derval Morris, National University of Ireland, Galway. What's the limit of detection of your test directly from clinical samples? And also, do you think it could be applied to environmental samples such as water?
Could your test be adapted to look at water?
If you mean directly from the sample, or if you grow the colony and then work on with these colonies, of course, yes.
From the actual sample?
I think you can do that as long as there is no other inhibitors or proteins that will decrease the pH value. I don't know exactly what kind of sample you're thinking about, but...
Sounds like you have a research project in mind.
Yeah.
We do. We have a poster we're presenting now at 11 o'clock where we've looked at waters for antibiotic-resistant organisms.
Using the NP assay?
No, not using the NP assay. That's the reason for my question. And we're particularly interested in virucytotoxigenic E. coli, and we've been looking at antibiotic resistance in that because it's a major problem in Ireland. So it might be interesting if you could apply this test. It would be a lot quicker.
The wonder of microbiology, the ever-changing field, because the microbes are still always smarter than us.
Yeah. And not only the environment, but my vet colleagues in Europe,
veterinary medicine,
they're all very much interested in that, because they cannot all afford these molecular testings, and so they would be very happy to have that kind of test. So they're starting now a project on this.
You've been watching ASM Live here at ICAAC. I'd like to thank my guests here, Dr. Laurent Poirel from the University of Fribourg in Switzerland, and Dr. Karim Morey from the Oregon State Public Health Laboratory System for their provocative discussion here this morning on antibiotics and new testing for those antibiotics in our microbes. I'd like to thank the audience for their participation in this edition of ASM Live. Please join us again at 11 o'clock Eastern for our next edition, where we're going to be talking about fungi and looking at the whole issue of fungi and antibiotics as well. Please join us again at 11, as I said, or listen for me, Michael Schmidt, when I join my colleagues Mozilio Schechter from Small Things Considered, Michelle Swanson from the University of Michigan, or Vincent Racaniello from Columbia University, when we all get together for our next edition of TWIM. This week in Microbiology, the podcast about the unseen life on planet Earth. Thank you, everybody, and we'll see you again.
Thank you.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.